Start when you're ready.
Okay, thanks, Rob. And yeah, I can't see the chat window, so questions I will have to read afterwards. So as Rob said, most people know me as the operations area coordinator. I've been with the OSG for quite some time, and recently was asked by a local researcher at the School of Medicine here at Indiana University to look into his scientific workflow. So I subtitled this presentation "How I Learned to Stop Worrying and Use the Grid," and I think I'll explain why that has that subtitle. So as we go forward, so the story leading up to January 2013, really OSG and IU have been close partners for a long time, but mostly based on the operational infrastructure, not on sciences actually utilizing the OSG from IU outside of the Atlas experiment, who's been a long time IU standing. But really, as far as other local researchers using the OSG, it hadn't happened. So we had always done the things that it takes to run the OSG, really looking at the support and the services, making that 24/7 support possible, running the critical services that allowed information to be shared, things like BDII and MyOSG, which were used for resource allocation by a lot of the experiments in OSG, and several other operational services, really the infrastructure that it took to run the OSG, the topology database and the certificate issuing, ticketing, and the system behind that, and then several other items, accounting is along there, and the GlideIn WMS. And the other thing that we did quite a bit was work with operational components, or interoperational components, the things that we shared information with other grid structures like EGI and WLCG as far as monitoring and accounting and information systems. BDII was also shared. So as the alternate title of the presentation suggests, really our job at Indiana University was worrying about the grid infrastructure. So we worried about OSG a lot, but not necessarily as users or as worried about it from our scientist or researcher point of view. So this all really changed. I'm sorry, got a little bit ahead of myself here. So we did anticipate this possibility back in 2010 and established the CSIU VO. And this was for really two reasons. So we could bring researchers on board from IU when they were interested, and have a virtual organization, a home for them to run on national resources if necessary. And secondly, to put in a support structure around campus resources that would want to be part of the national infrastructure. And we've had some success in part two, but not really in the first item there. And we have both the Atlas cluster, obviously, are part of the Atlas cluster, Midwest Tier 2 cluster located here, and then we have some other infrastructures. The Quarry node is online, and we're working with Mason, and we've recently brought our School of Informatics, one of their clusters, online. So we had some bit of success in bringing resources that wanted to make their resources available to the OSG, but not much as far as the actual researchers running. Now, as of today, I actually looked to see what the CSIU adoption is 3 years later, and I really saw this on only about 20 resources and at about 7 institutions. This means that a VO, and we did no real pushing to have this VO adopted, but through natural courses, a VO that is established 3 years later is only really used on about 20, maybe even less than 20% of the OSG resources. So this, as I'll explain later, became a bit of an issue. So in January, we were approached by the IU School of Medicine, and we were approached by them because they previously had an exceed allocation, and this exceed allocation ran out, and they put in a new proposal. Really, they put in a proposal that was asking for a bit too much, thinking that the review board would then downgrade the proposal and give them some portion of that. However, it was actually flat out rejected, and this was for a few reasons, not necessarily that the science was bad, but more for the reason that the proposal was done half-heartedly and the resources being asked for were far greater than they had asked for before. So really, the scientists came to us and said what I put in here. It gave us a box with an executable, about 3,900 receptors, about 500 ligands, and some conversion tools. And this was handed to us saying, basically, this is what we do, can you help us out? And we thought to ourselves, of course, with all those things, we should be able to have a pretty good time here on the OSG. Something to keep in mind about both the executable and the data files is these were very small. The executable AutoDocVina is about 1.8 meg, and the data files for both the receptors and the ligands was only a few kilobytes, so very small amount of data, which took a level of complexity out of it, as I will show. So the protein docking was done for this SPLINTER project, which is a interactome of basically dockings between proteins and ligands. It uses a executable called AutoDocVina, who's basically used for drug discovery and molecular docking, and this is widely used open source piece of software and heavily used in the biological sciences and people studying medicines and how they potentially one chemical docks with another chemical. And our first run, we basically, as I said, had about 3,900 proteins and 5,000 ligands. Each protein needed to be docked against each ligand, and that would have given us a total of about 19.5 million docked pairs. And the other thing to keep in mind, these are very short jobs and very serial. They only need the data that one of each of the proteins and ligands, and they lasted about 3 minutes when you did the full docking. So each job, on average, lasted about 3 minutes, so relatively short and very, very serial. So we had some preliminary barriers that we had to overcome. As I mentioned earlier in the CSIU slide, it was not very widely adopted. In fact, the 20 sites that I mentioned about, or the 20 resources that I mentioned about, a dozen of those were actually at Fermi, and we could not use that for the fact that we wanted to use a GlideIn, and Fermi at the time didn't accept GlideIn jobs. So even of the 20 resources that we had, about a dozen of those were unusable at the time. So we decided to use the OSG-VO and the OSG-XSEDE head node. And this allowed us to access all resources that the OSG-VO was running on, which is a much wider spread than the CSIU-VO. Of course, as I mentioned, we had more than 19 million jobs initially, and negotiating each of those jobs individually would have been quite difficult, and would have spent a lot of time in the negotiation phase and the matchmaking phase rather than in the job phase, making the run be a lot longer. So we chose Pegasus as the best option for doing some of the workflow management, and I'll talk a little bit more about that in a few slides. And the other thing is, of course, we had the data handling with 19 million plus small files, which were all outputting to the same place. This ties up a file system, as you can imagine. So after we saw the barriers and found solutions for at least the first two. The data handling is still one that is clunky and takes a little bit of time, but after we did that, we prepared the run. So what we did was developed a wrapper around Autodoc Vena. It does the things you would expect from a wrapper. It checks for the proper arguments before executing and fails if it doesn't have the proper arguments. It executes the Vena script, converts the output from one format to another. This is another executable that moves it from two commonly used formats. Of course, the database that we were trying to get it into at the end was MOL2 format, so we had to do a conversion from PDBQT to MOL2, and then it cleaned up all except the output file. So it did some housekeeping when it was done. And then initially, a Python script was created that made a Condor submission file. And in this Condor submission, as I said earlier, we have small executables and data, so we sent the executable and data with each job. One very important aspect is we had the identifier of the project name as CSIU. This allowed us to do the accounting properly inside of the OSG VO. So we were still seen as CSIU even though we were operating in the OSG VO. And initially, as we were doing testing, we did testing with each of these jobs individually. So we hadn't initially handled the issue of grouping or of negotiation, which I mentioned earlier. So as we were doing this, Mats Ringe from ISI got involved and decided to help us out with the grouping problem and creating sub workflows, and he then hooked us up with Pegasus, and in fact took over a little bit of the work on the project in the Python executable and added Pegasus in to do the workflow management for us, which was, again, it helped out in a few ways. It helped out with the gathering the inputs and making the workflows, but then also with grouping these together into larger jobs. Of course, we didn't want to submit 19 million individual 3-minute jobs, so we grouped them together into about 25 jobs per, or 25 jobs per sub workflow. This made the job last approximately, if you have 3-minute jobs, 75 minutes or a little over an hour, which Mats informed us was a sweet spot, was somewhere between 1 and 2 hours on the OSG. So that allowed for, if we got some longer jobs, to still complete within that time. And it also only sent the executable once per 25 jobs and also grouped them into proteins. So while it had to send 25 separate ligands, it only had to send one protein with each job, which helped out quite a bit. I'm showing the Pegasus slide here. I'm going to read Matt's comments on this slide. I'm not a Pegasus expert by any means, so Pegasus questions I would prefer be directed towards him. But Pegasus is a general workflow system which can handle small, simple workflows all the way up to workflows with millions of tasks and complex task dependencies. The advantage of using something like Pegasus, of course, is that it provides a high-level description, language for your computation, and the workflow is planned beforehand and compiled into an executable workflow. There are several advantages, one being that during the planning step, Pegasus applies optimizations, clustering, and data management is included with that. And then, of course, there are portability and reusability issues that Pegasus addresses, and these workflows can then be planned for execution on different infrastructures, campus grids, clouds, and such. And one of the things that was quite nice is when Pegasus completes, it offers some tools for statistics and for monitoring of the workload during the run. and debugging, even though we had very little actual debugging in this run. The first run actually completed without much errors, or at least with an error rate that was so low that we could re-execute very easily. As I said, it provides some statistics and graphs at the end of the workflow, so you can see how the workflow run. Let's see, and one of the things, as I said, that was quite nice for us was this sub-workflow and making this hierarchical workflow so we could watch the progress of each sub-workflow along with the progress of the entire run as the run progressed. And as I said, all the short tasks clustered together in one. It got us out of that negotiation issues which we had seen, or which we expected to see if we had submitted 19 million individual jobs. So as I mentioned, data output, we really didn't find a good solution. We did the initial data output. We did some semi-manual steps to move it from the output directory to the database. Later we wrote some shell scripting, still pretty inefficient, and as I put here, I'm happy to take suggestions from anyone who has handled a large number of files, outputs, and moving them around in data management with a large number of files. Even though they weren't particularly large, it was a ton of them. So what was the outcome of this? So this was all then published to the website www.biodrugscreen.org, and this is where the stuff lives now. Sammy Marroway is the scientist who recruited this. I would suggest anyone who wants to see the details of the science to visit the site. It has some explanations of it. And what it does is basically simulations of dockings. This is one example and rotations of that example. This is a zinc docked with a sicillin. And the yellow portion that you're seeing in these docking are where the two molecules will share surface area. This then is important for simulation and later reconstruction. A biologist, I'm told, can look at this and determine whether this looks to be a good docking or a bad docking. There's also some automated scoring tools. As you can see on this slide, from the table on the left, there's a ranking table as to which chemical binds best with or shares the most surface area of docking with the protein that you're testing on. So real short explanation of that, as I said, simulates the optimal surface area for a drug to a protein. After the simulation is complete, which is what the AutoDock is doing, each docking is scored and ranked. Now we're not doing the scoring and the ranking as part of our sub-workflow yet. This is something that we've mentioned to the School of Medicine and something that may be worked on in the future. Right now, they're just happy to be getting the simulations completed. So the website, and I'll go back here, not only offers the visualization and the ranking, but it also offers a link that you can order this drug if you find the ranking in the simulation something that you want to test. And really what this does is moves the drug more quickly from a simulation phase to a wet lab testing. Someone can go here, put in the drug they're interested in, see the rank that it has with various different proteins, order it, and start going to the wet lab quicker. So it is quite a bit of speeding up the process that it takes to get drugs into testing, which is quite neat. So the first 1 million jobs, we actually did a distribution here, and you can see from this relatively simple graph the usual suspects, I think, are in here, along with a few not so usual suspects. we got a lot of work from Nebraska and Wisconsin, as you would expect. However, I didn't actually expect to see so many jobs come back to IU for execution. SMU is one that also provided quite a few cycles that I was surprised by, as their name doesn't appear in the, or at least before that hadn't appeared a lot in the accounting graphs that I was watching from a operational standpoint. So other stats about the initial run, this was the first 19 million jobs we did, about 1.4 million hours. The graph on the left, it's the yellow portion, so it ran over a period of about a month, completed those dockings, and we were able to move that data out. Since then, we've done a couple more runs for them, 2 more runs, and now we're up over 30 million dockings that we've done, and CSIU is green in this one, actually, so over 3 million hours of time, and you can see this comes in various different runs. We're the green in both these graphs, so you can see the work that was done in January, February, and then the other runs were done in the starting with April to mid-March. So just a few things about the handoff, and this was the big success story, I think, in the entire process, is we were able to hand this off to the School of Medicine grad students so they could then do this on their own, and they don't need to call up OSG or CSIU. So really they have 3 simple steps. They put the data in the place that it can be found, point the configuration at the data. This is the receptor directory, the ligand directory, and the executable that changes things from PDB-QT to MOL2, and then hit the submit script. And again, this highly leverages Pegasus for creating the sub workflows, but these are 3 simple steps that we can really give to a grad student who is studying biology, and there's not a whole lot of learning that they have to do about Condor or the grid in general. They basically go and update the numbers or the directories in this file and hit the submit button. And of course, that's the big red button that sends them on their way to getting their job done. So,
okay, Rob, very nice.
That's a general explanation.
So Miha has a question here in the chat window. I don't know if you can see this,
happy to share some more of the
but
technical,
he's asking if the medical researchers at IU are happy. If yes, do you collect what they say?
Overview here today. If anybody's interested in seeing any of the actual scripting that's done in Python or the shell scripting that was done to make this happen, I would be happy to share that with them. And I will open up for questions. So they are happy. We don't collect specific quotes. Now we have collected a few things here. One is we had a presentation in the scientific research section of the all hands meeting, and there is a ISGTW / OSG research highlights paper coming up. So we've sent our communications people to talk to the medical researchers. So not directly. I think really the anecdotes I would say is that it really was quite interesting to move from looking at the grid from an operational standpoint to moving to looking at it from a user standpoint. And I think I learned more in the first 3 months of actually looking at things from a user standpoint than I had in the, well, at any given condensed time. I won't say over the entire 10 years that I've been with OSG, but this was really a packed period of productivity and opened my eyes to what was going on to the OSG. one other thing there is that I'll say, and then I'll answer Derek's question, is that really having taught several grid schools, my knowledge of the submission and stuff was really learned from knowing the material in the grid school. So the new revised grid school that's been taught for the last 2 or 3 years in University of Wisconsin really contains all the base knowledge that you need to do something like this, which I think says a lot for Tim Cartwright and Alan Roy overhauled that program. And the students I know are up there next week, and I think, as I said, the vast majority of what was needed to do this is available to students in the grid school. So the most difficult part of the process, Derek, it really is handling the output of all those files. I think writing the initial scripts and the workflow was not so difficult, at least to the point that we got Pegasus involved. And I can't really say how difficult it was to add the Pegasus hooks in. I can say that Matt did it in less than 8 business hours.
So that was one part I wanted to go into a little bit more, Rob, which is the output from each job and getting them back to where they're needed for the database. And I didn't quite get exactly if you were trying to use a storage element as a target for the output, or how were you pulling them back to the place before they got uploaded to the database?
So we actually downloaded them to a network device here at the GOC, which is a tool that probably most people won't have accessible on the OSG. So we kind of used the fact that we had a network device available for the OSG services that we could put this data on.
Okay. Other questions?
Off of the execute node. And so
Derek is typing something here. While he's typing, let me offer another question, which is with the grad students gaining access, did you just set up accounts on the OSG-XSEDE node for all these graduate students, or how many accounts did you set up?
Yes, it was 2 accounts. There were 2 grad students working on it, and they had accounts on some of our local nodes. They actually were giving us, they were putting the data in place prior to us doing this, so we extended that to the OSG, the XSEDE OSG submit node. So they do have accounts there. They have given us their keys. They can log onto there directly and start this workflow. And then to answer Derek's question, yes, these will be ongoing. Usually they come in a set of somewhere between 10 and 20 million jobs. And at this point, when they are ready to start a run, the grad students can press that button, which is quite nice. And I expect to see new runs starting up at any time. They, of course, get these 19 or somewhere between 10 and 20 million jobs. They do some amount of scoring on each of them and then prepare the
Another
next
question from Derek.
run submission. I don't have an exact time on that yet. It really depends on when they clean up from the previous run, which I think ended in late May, if I'm correct, and if the accounting is correct. So it ended in late May. We also are dealing with grad students who do have a summer that they sometimes take time away from things. Yep. So the portal, we are working with Marlon Pierce and his OSG, Open Computing Grid Environments group, to do more of an on-demand type submission. That's still in the future, but the hopes is that, of course, we can populate this interactome to the point that most simulations are done when the researcher gets there, and they're as simple as returning the file. However, we do want a mechanism that for those who haven't been simulated, somebody can hit a button on the web portal and get that specific one submitted. That work's been discussed, and but really activity on it is still very, very early. I would say probably no actual coding has gone into that. And also previously, as far as the web portal, previously it did submit to...
Another question there from Derek.
And we do have a startup allocation on XSEDE for that portal. So they have 200,000 hours for that on-demand service, which is still working.
So Rob, why don't you repeat the... repeat the question for me.
Currently with the bigger batch submissions when they have 2,000 of these ligands that they want to test. Yes, the allocation is on OSG. So Kimbasco, yeah, okay. So Derek asks if the XSEDE allocation is for OSG, and yes, those 200,000 hours are for OSG. So I guess that really answers the question before, is that yes, this portal can and will be connected to do on-demand work on the OSG, but through XSEDE mechanisms. And can Bosco play a role behind the portal? That's a good question. And we're also working with the bioinformatics group here for submitting BLAST jobs. Anybody who's familiar with bioinformatics knows what BLAST is, but it's basically a comparison of sequences to various different...
Another question on the way. While Mia's typing, does anybody else online have a question?
Play a role between the Galaxy portal and the OSG submission for those BLAST jobs.
Okay, that's a comment.
But as we see it now, we don't anticipate Bosco being involved with the batch submission process, though the portal is a good question, and one that we might want to suggest have a conversation with Marlon Pierce. And as I said, just one final comment. If anybody's interested in the more technical details, I wanted to give an overview here and not dig too far down into it. I would be happy to share all of the work on if someone wants to see the scripting that we've done to make this take place. Yes, we plan on publishing this work with Sammy Merroway. So Derek asks if the work will be published. So two things. One, we already have a logo on the web portal, as the people visiting it have seen. And I'll bring this back up for anyone who says this. So we have this Open Science Grid logo. available. But yes, Sammy does plan on publishing this work, and I've already talked to him about making sure that OSG and CSIU and the people involved get properly referenced.
Okay.
So there should be.
All right. So I want to thank Rob. Sounds like we're out of questions here. So I want to thank Rob again for preparing a nice set of slides and offering a lot of interesting insights as to the work that he's been able to do and leverage there at IU. I think this is great. So just for next time, we have in about a month, we'll have a